as Olivia mentioned, my name is Lauren Walk, and today I am presenting my senior thesis, as she said, titled Manipulation of Iron Oxide Powder Suspended in a Hydrogel. So a little agenda of what I'm going to go over today. I'm going to talk about background information, the current study that I conducted, the results and discussion, limitations of the work, as well as future work. So a little background information on what inspired my study. These are a few techniques that I looked into. So the first is dealing with bioprinting. This can be defined as assembling and patterning living and non-living materials to create structures for use in medicine and biology. As you can see on the bottom left, there's a picture that shows the principles of bioprinting. So there's a bioprinting nozzle, which prints out the different cells and it builds it up into a 3D structure, much like 3D printing if you're familiar with it. Also, there's magnetic levitation that I looked into. This deals with binding cells to magnetic nanoparticles to render them magnetic. So then an external magnet is introduced and then it will levitate the cells. So bottom right here shows a picture of that. It has the cells in the petri dish and then a magnet is placed on top. So it creates a cluster of the cells when they're drawn to the magnet. So this 3D structure of the cells creates a better way for scientists to look and study these cells. So then these two principles can be going into tissue engineering, which can be described as growth of tissues, organs, or cells, and to produce organs that can be implanted into a host. So these two techniques can be used to engineer tissues. So some of the applications that are being used today for these techniques, as I mentioned, tissue engineering, you can use bioprinting or magnetic levitation in order to make these tissues and structures. And also drug delivery, you can implant the magnetic cells and make the drugs magnetic. That way you can get them to wherever they need to go into the body to be most effective. But some limitations of what's currently being done is that there's limited control of the cells or particles. If you're just using one magnet with one magnetic field, cells are just going to go to one place. Also, limited types of shapes or structures can be made today. Mostly it's just a sphenoid or a ring, which would be helpful for making like a vein-like structure. The image to the left shows an animation of the cells with one external magnet. So they're all drawn to the magnetic field and create the sphenoid. So this is a really beneficial and upcoming thing that's happening right now. But more control of what can happen is important to be looked into. So my current study was to control and manipulate the movements of magnetic hydrogels with varying magnetic fields. So to do this, I constructed two solenoids to work in conjunction with each other and prepared sodium alginate hydrogels with varying amounts of iron oxide powder within them, and then tested different magnetic fields with different concentrations of the iron oxide powder. And this was to determine the ideal conditions of the magnetic powder as well as the magnetic field to see what would be best to control these hydrogels. So the first part of this study dealt with the solenoids. A solenoid, or also known as an electromagnet, is just a coil of wire and a current passes through it and then it renders it to act like a magnet. So for mine, I used a 1-inch diameter iron core rod, which the wire was wrapped around. This iron core produces a larger magnetic field, so it was a stronger magnet. And also I used a 16-gauge copper wire, and each of the solenoids had 30 wrappings and 3 layers for a total of 90 wrappings. Now when constructing a solenoid, the gauge wire and the number of wrappings determine the strength of magnetic field. So that's why I chose those particular specs. Now to make them act as a magnet, they needed to be hooked up to a power supply. The power supply I used was 30 amps with variable 30 volts. And as you can see, that is the setup of the small circuit. So there's a terminal board there along with a 100 ohm resistor in order to keep the current down, keep things at a safe level. So the solenoids were placed 3 inches apart from base to base, and when it's powered, the magnetic field is right in the center there. So in order to measure the magnetic field, I used a Hall effect probe. So the probe went in the middle between the two solenoids, and then I measured the readings from 0 volts all the way up to 30 volts. And the initial trials of this, the Gauss readings, which is a measure of magnetic field, did not seem to be high enough. So I did some adjustments to the solenoids. That way I would be able to hopefully get movements from the hydrogels. So now each of the solenoids have 30 wrappings and 7 layers, so a total of 210 wrappings in each of the solenoids. The next part of the study dealt with the hydrogels. So sodium alginate is dissolved in erythrocin B, and then also the iron oxide powder was added into that solution. This solution is then drawn up through a blunt tip needle and then extruded into calcium chloride. Now when it's extruded at a constant rate, it creates these little beads. As you can see, the black little beads are the hydrogels. And I added varying amounts of the iron oxide powder in 5 different concentrations, from 0.75 grams all the way up to 1.75 grams. So the next part of this study was to combine these two components together. So a beaker of the hydrogels was placed in between the two solenoids that are again 3 inches apart. The power supply was then adjusted from 0 volts all the way up to 30 to see what would be the best to make movements in these hydrogels and control them. Unfortunately, there were no movements from any of the particles, from any of the concentrations, any of the magnetic field levels. So a new direction had to be taken. So to do this, I concentrated more on what was happening today. So I used a neodymium magnet, which is a rare earth magnet that's very strong, and placed it varying distances away from the beaker. It's supposed to be a video going, but technology, you know. And it would show all of the magnet going up to the beaker, and then all of the cells go to it rapidly. And this is the highest concentration, so the majority of them move to it. And this was, purpose of this part of the study was to determine optimal distance and concentration of the particles to create the best movements. So when I did this, I measured each of the concentrations and distances away, and then figured out the percentage of the gels that moved. So this is a chart showing each of the concentrations of the powder along with the distances away of the magnet. And the red line, which is the highest movements, is 1.75 grams of the iron oxide powder in the hydrogels. So this isn't really a surprise that this was the highest movement, since it is the highest concentration. So limitations of my study, dealing with the solenoids, there were material and time constraints that didn't allow me to further explore into the solenoid to get it to work to deal with the varying magnetic fields and try to get movement from these hydrogels. Also, I was unable to determine the magnetivity of the iron oxide powder, which would have been helpful because then I would have known exactly the magnetic field reading that I needed and better prepare my solenoids to see what they would really do. And then also with the hydrogels and the magnet, time constraints, I only allowed to do 1 testing with the magnet and the solenoid, or the hydrogels. Ideally, I would have liked to have done 10 trials of each concentration at each distance. So since I wasn't able to, this gave me limited results to work with. Future work to work on, improve the solenoids. Either do more wrappings of the copper wire or use a smaller gauge wire, both of which will result in a higher magnetic field and hopefully be able to control these hydrogels. And also perform more trials with the bar magnet and the hydrogels to obtain more accurate results and really determine more concrete correlations between the distances and concentrations. And potential applications of this work, as I mentioned before, drug delivery, determining the amount of concentration of magnetic particles to put in drugs in order to manipulate where they're going to go inside the body to be most effective, and also development of more complex structures. So like I talked about, the sphenoid and the ring, trying to make something in a more concentrated and controlled manner than just those. All right, I thank you for coming today, and I will take any questions at this time.
So in your paper, you mentioned the potential applications. You said things like manipulating structures to make veins. You also said drug delivery, which we'll look over here.
Yes.
You also mentioned things like cancer treatment and stem cell research. Could you explain, if you could, how exactly that would be
So
applied?
what they're doing is much like the magnetic levitation. You can take the stem cells and expose them to the magnetic nanoparticles, which will then make the stem cells magnetic. So then this way they can take the stem cells and structure them into whatever type of like organ or structure that it needs to be formed in order to make whatever structure they want, whether it's like a vein, so then you would make more of a ring-like structure, or a heart to make like the sphenoid. So that's kind of that sort of deal of what that deals with. Thank you. Yes?
Do you foresee any application in the controlled release field in drug therapies?
I think definitely this would be a great application dealing with the controlled release of drugs and seeing what rate it is released, and then also being able to control where it's going with the amount of release. That makes sense.
Yeah, the reason why I'm interested, our daughter-in-law is actively involved in Frankfurt at the university
Okay.
in that area.
Okay, thank you. Is that it? All right, thank you, everybody.